excited. I'm really excited to have two of my interns be presenting their work today. The first up is Natalie. She's a senior at Oregon State University. She's a microbiology pre-med major. Now, since she's microbiology and I study the microbiome, I was kind of anticipating that she'd be interested in working on some of my microbiome pipelines for analyzing the sequencing data, but she actually really wanted to understand and get involved with clinical research and recruitment. So her project really focused on trying to mine and analyze the recruitment data that we had from my previous studies, and also getting some exposure to the clinic at the Center for Women's Health here at OHSU.
All right. Okay, so good afternoon, everyone. Thank you for that introduction, Lisa. So today I'll be presenting on data-driven shifts in clinical recruitment strategies. So just an outline of my talk today. First, I'm going to give an introduction to overactive bladder and the general participant involvement in this study, and then talk about the recruitment in the initial study, and then building off of that initial study recruitment, how that guided the recruitment change in the current study that we have going on right now. Finally, I'll talk about the future steps in this study and my internship takeaways. So overactive bladder is characterized by the detrusor muscle in the bladder squeezing too powerfully or too frequently, resulting in urgent and frequent urination. This is really important to study because it's highly prevalent in both women and men, especially in older ages, and also it has a high correlation to stress and anxiety. This stress and anxiety results in an effect on the quality of life in many people, especially regarding limiting socialization due to restroom availability. Also, overactive bladder is not well understood, mainly because its causes and its treatments aren't well developed. In this particular study, we focus on postmenopausal women and have three groups that can be categorized into. Those three groups are overactive bladder wet, where leakage occurs from urgency and frequency, overactive bladder dry, where leakage does not occur from urgency and frequency, and then the control group, which experiences no overactive bladder symptoms. This is an overview of participant involvement in this study. So initially, a participant is contacted, either contacts the research unit on their own or is contacted by someone in the study. Then their eligibility is assessed via a phone screen. Then, if eligible, they would be scheduled for a study visit, their samples would be collected, and their data would be analyzed, and that participant would be enrolled. My big question for this project was, how can we use past recruitment challenges to guide our recruitment strategy in this current study? I attempted to do so by addressing these 3 points: recruitment documentation challenges, community reach, and participant retention and interest. So recruitment in the initial study used many referral sources, some of these being online, different flyers, Facebook, and then some provider referrals. Recruitment was documented in an Excel file. This was initially made to be accessible amongst the study team, but also serve as a source of communication for the study team, which had multiple people doing recruitment. But similar to the EHR, because multiple people were implementing and these had free response cells, we had lots of different interpretations and lots of different verbiage going into the Excel file, making an unstandardized source, which created many different fields. An example of this was it had 14 enrollment sources and 68 referral sources, as seen on the right side of the screen here. This inconsistent data entry makes participant data really difficult to track, first of all because of the referral sources that came out of it. As you can see on the left, there are many different variations of just flyer, some of these capitalization errors, some of them having locations, some of them not. But also it made it difficult to track which contacts were actually enrolled. So we had one file for people that were contacted and then one file for who was enrolled in the study, but we didn't have a way to map those two together. And then finally, we were interested in at what point, if any, did a participant lose interest in being involved in the study. Going forward and referring to this right-hand side list, which is not exhaustive because this is not all 68 of the different referral sources that were found, we wanted to consider capitalization, but also too general of statements like online, and then also too specific of statements like names of study coordinators or friends. Because the spreadsheet was not standardized, plotting the referral source against the enrollment status gave a really messy and illegible plot, as shown up here on the top left. So then we went through this data cleaning process to produce a more informative and more usable plot here. However, cleaning and tidying data is not as easy as just sweeping it up with a broom, so you have to consider what tidy data actually is. So tidy data has, each variable has a column, each observation has a row, and each value has a cell. To do so, you can use various R packages like tidy, data table, and dplyr to join subset data sets and get out variables that you need. But also, like you saw in the earlier figure, we needed to standardize those names so you can use levels and character strings to turn all those little f's into flyer. However, when you're cleaning the data, you need to have a goal in mind. For example, if you were interested in those locations that those flyers came from, you might not change all of those little f's into simply flyer. You might keep accounting for the location in which they were hung. But this is just a summary of that initial data cleaning regarding the referral sources and the enrollment statuses. So in the unclean data, we had 68 referral sources, which turned into 14, and then we had 14 enrollment statuses, which turned into 7. In addition to those tidy referrals, I mentioned that you do have clear enrollment statuses after cleaning that data. So not surprisingly, not all individuals who are contacted at the beginning of this workflow end up being enrolled in the study. That's just the case with clinical research or research in general. But I wanted to pull these enrollment outcomes with the hope of depicting where in this sequence people lose interest, but that data was unfortunately not available to determine exactly if it was after contact, after eligibility, or after their study visit. But from these different options of loss of interest, you can kind of infer where they might be. The loss to follow-up one is a bit ambiguous. That sounds like it was after contacting the patient after they expressed interest the first time. And then there are these not interested categories due to specific reasons, the biggest of which was the catheter. In this study, we are interested in the microbiome in the bladder, and so we used a catheter to obtain the bacteria in the bladder without contamination, and a lot of people were uncomfortable with this procedure. Other options of not being interested was the trip to OHSU, compensation, or just other things going on in their lives. Another part of not being interested in the study that we can't really do much about is being disqualified from the study, whether that be having an existing condition that makes them ineligible, or being on antibiotics, which in that case we have to revisit them later on in the study, see if they're eligible then, and that follow-up sometimes results in them not being interested any longer. So a question that I had going forward, just because we didn't have the information on exactly when the referral lost interest or the participant lost interest, I wanted to know if some referral sources have a higher retention rate than others. So to evaluate this community reach and also retention, I made this plot of passive and active recruitment approaches. So passive recruitment involves all sources that are out in the community or that a participant sees and contacts the research unit on their own, where an active recruitment is a direct targeted approach where physicians or study team or any other option that the participant is identified as a possible participant and then contacted by the research unit. So in this case, our passive recruitment included flyers, newsletters, Facebook, etc. And our active recruitment included the EPIC cohort MyChart messages and then also provider referrals. Based on this proportion plot, it's not very conclusive to decide whether or not active recruitment does have a higher retention for participants, though it is only 0.5% higher on this plot. But past literature does suggest that direct targeted approaches end in higher participant retention, and also we have this unique opportunity to begin recruitment in the Center for Women's Health, prompting the study team to take advantage of that extra active recruitment here. So this leads me to the current study recruitment based off of that initial study insight. So in the current study recruitment, I still wanted to consider those 3 main points where that recruitment documentation to standardize the data was really crucial. First of all, that log in Excel was standardized by adding drop-down menus, pretty simple, but it exponentially improved the standardization of this recruitment log. Also, we started using this database called REDCap, which can hold all of the questionnaire data, all the study visit data, and can be easily pulled from, so you get subsetted data frames without having to look at thousands of variables at one time. Also, with this community reach aspect, we wanted to continue to utilize passive recruitment platforms, mainly because they were effective, and also it has little user or study team time in going out into the community. Also, we wanted to begin this recruitment in the Center of Women's Health just to explore that impact of that active recruitment. And then also, by doing so, we identified participants in the CWH that could be eligible via EPIC, and then talk to them in person about their concerns or their interest in this study. Through that, we wanted to explore the efficacy of the participant interest and retention in CWH. This is a general workflow for the CWH system. So first of all, we had that participant recruitment, which involves that initial contact. Then the data was entered and verified in REDCap, that big database that I talked about earlier. And then we cleaned data with different R packages, as I mentioned. And then we visualized and analyzed in RStudio. This workflow may look simple, but it's really important for reproducibility of this work. Recruitment is ongoing even after my internship is done. And so at that REDCap level, or even at the participant recruitment level, new files can be uploaded into this workflow and ran to have a similar analyses. So just like you've seen before, we have this dirty data and we have this clean data. But even looking at this first plot of the dirty data, this comes from standardization of the log to output a much neater, still readable file as compared to the completely unreadable one that we saw in the initial study. And we go through this same tidying data process to output our clean data file to show us our referral statuses versus our enrollment statuses. So to zoom in on those 3 most common referral sources, we look at CWH Clinic, Facebook, and newsletter. You can see those here. CWH, Facebook, and newsletter were the 3 most common. So just for reference, this plot on the left-hand side is before my internship, so that's 1 year long of time. And then on the right-hand side is that 10 weeks that I've been here from late June to now. It's also important to note that these large blue sections are disqualified or not interested portions of that recruitment. Unfortunately, this is just characteristic of clinical data or clinical research, but it's emphasizing the difficulty of participant retention. So just zooming in a little bit more into just the Center for Women's Health section, this plot also shows enrollment and referral based on contact time. So on the left, this is actually a 5-month period because they didn't begin CWH recruitment until February of 2019. So this is from February to late June, and this is from late June to now. The difference between these 2 plots is just the time spent in CWH and the emphasis on this active recruitment. So on the right-hand side, or the left-hand side, not very much time was spending per week, and the appointment flow or the appointment volume wasn't really being considered. But on the right-hand side, I would spend 4 to 8 hours a week in the clinic, carefully looking at when there were many appointments or many patients to be seen. Waiting around in CWH is a large time commitment, especially for people that have other study recruitment efforts going on. And so if I would try to go during times, I would have 12 patients in like 4 hours versus 2 patients in 4 hours. And that makes a really, takes advantage of the time that you have. Additionally, I wanted to note that because my internship is ending, I am unable to fully decide whether this big pink area shows that the CWH or increasing CWH recruitment made that great of a difference on overall retention, because this middle darker pink area is still patients that are in follow-up. So those participants are still able to become not enrolled or enrolled in the study. Building off of that, this plot here on the right is again the participant retention plot with proportions versus active and passive recruitment. So that the two bars on the left, those are enrolled versus not enrolled for the year portion or for all of the recruitment from a year ago until now. And until this darker pink in the center becomes either enrolled or not enrolled, we're unsure whether this active recruitment makes a huge difference or not. However, as you increase participant reach or community reach, you're getting more contact with patients and you have a higher probability of participants being enrolled. To go back to this recruitment documentation, for the future, I would advise continuing to standardize the documentation depending on what your goals for your research are. So for example, earlier I mentioned that I would like to know where along that recruitment line participants are no longer interested. So maybe taking those or making more specific measures on that. But obviously you can modify that Excel log as needed and still keep it accessible to all the study team that would need it, and it's relatively easy to use. Also, for community reach, I would utilize passive and active recruitment continually. First of all, because this Facebook and other online or newsletter sources are really effective and they reach a larger amount of people at one time than talking to people in the clinic. But talking to people in the clinic does create that sense of community, and they can get their questions answered and concerns put to rest right there, versus an online source, you're at risk for trolls or people that don't fit the eligibility standards and so forth. Lastly, participant interest and retention, because we still have so many in this in follow-up stage as this study is ongoing, I would love to revisit this and then have a greater sense on whether this active or passive recruitment ends up to be favored over one another. So overall takeaways from this research time, I really dove into R as a data cleaning and statistical tool, something that I had a little bit of familiarization with, but not a lot. So I was really excited about that, and I feel like I accomplished that goal. But then also, I am a pre-med student, and seeing that clinic flow and getting that exposure to how physicians operate and how how patients are perceived in the clinic was a really cool experience. And also, just in general, valuing documentation and reproducibility. R Markdown was great for this because I learned the value of annotating my code so I knew what I was doing when I revisit it. And then reevaluating, potentially restructuring your methodology for that same reason, going back and seeing how things maybe didn't work how you thought they would. And then finally, taking advantage of resources, whether that be Dr. Karsten's, my fellow interns, MAs or physicians in the clinic, or just my friends around or experts in the room. Finally, I want to acknowledge Dr. Karsten's for taking our interns on this summer, and then the DMICE program for making this all possible, and the National Library of Medicine grant. Thank you. Okay, thank you.
Getting set up, I'll introduce Celine. Celine's also a rising senior at Oregon State University. She is a bioresource research major and pre-med. Her primary goals for the summer was to learn more about human subjects research, as well as improve and learn the command line and also scripting. So she worked on my project that involves looking at how the brain regulates bladder function, and she's going to talk to you today about understanding the connection between the brain and the bladder and some of the exciting analyses that she did with processing the neuroimaging data that we've collected in my research so far.
Thank you for that introduction, Dr. Karstens. So I will be switching gears a little bit and talking about another one of Lisa's studies. And my work this summer focuses on understanding the connection between the brain and bladder and in subjects with urgency urinary incontinence. So presentation overview, I'll first talk about the urgency urinary incontinence study and then give a brief background on neuroimaging and then follow that with information on the human Connectome project pipeline that I used to analyze my data, and then talk about the adolescent brain cognitive development quality assessment, which is a protocol that we use for quality control. And then I'll talk about what the future for this project entails, and then ending with what I've learned this summer in my internship takeaways. So what is urgency urinary incontinence? So urgency urinary incontinence is a severe subset of overactive bladder. So women who suffer from urgency incontinence have frequency, urgency, and leaks. So that means that when they have the urgency to urinate, then it follows with immediate leakage. And this has high economic cost, and it's a burden on their quality of life. And about 19.1% of women between the ages of 65 and 74 in the United States have urgency incontinence, so it's important to study it. And the purpose of this study is to investigate how the brain and bladder connect, and we do this by identifying structural and functional differences in the brain between women with urgency incontinence and women with no history of overactive bladder. And our approach was to utilize structural and functional brain imaging techniques, as well as the human Connectome project pipeline that we process the data through to combine different modalities of imaging data to compare across subjects. So this is an overview of the data acquired for the study. So we have 36 women that are controls, and these women have no history of overactive bladder, and then we have 58 with urgency urinary incontinence. So we collected their age, BMI, and history of incontinence treatment, and if they did receive incontinence treatment, what type of incontinence treatment they received. And then we used validated questionnaires that were used to assess incontinence symptoms and their overall quality of life and pelvic disorders. So the International Consultation Incontinence Questionnaire was used to evaluate severity, frequency, and impact of quality of life. And then the Urinary Distress Inventory was used to evaluate frequency, urinary leakage, any difficulty emptying their bladder, and any pain or discomfort they felt in their pelvic area. And then we asked all the subjects to have a bladder diary that was self-reported, so we asked them to keep this for over a 3-day period, and we collected frequency, urgency, urge leaks, nocturia episodes, and fluid intake, and then this was averaged to find the average per day for each variable. And then lastly, we collected neuroimaging data. So we collected a structural MRI scan, a functional MRI scan, and the functional one was collected with the bladder filling protocol that was used to mimic the emptying and filling of the bladder. And there's 2 types of functional scans collected. One was a resting state one, and then one was a task-based one. And the purpose of the resting state scan was for a co-registration for transforming the resting state and task-based scans together, and then the fMRI scans was used to see how different brain regions react to the emptying, filling, or at rest. So this is some background on brain imaging. So I'm going to compare structural and functional brain imaging because those are the 2 that I used in this study. So magnetic resonance imaging uses strong magnetic fields to acquire images. So structural brain imaging shows brain structure, and it has high spatial resolution. An example of that would be the T1 weighted image, which is up there on your left. And then Functional brain imaging maps brain activity over time, and this one has low spatial resolution, and it uses blood oxygenation level dependent, which is bold, so it measures brain activity through blood flow. And this is because when there's neural activity, then there's an increase in oxygen consumption in that region of your brain, so your body will respond by increasing blood flow of oxygen-rich cerebral blood to that area of the brain. And then an example of this would be the resting state in task-based scans. So up there I have an example of a resting state functional connectivity MRI. It's a reference image. And then this is a hierarchical structure breakdown of data acquisition of MRI data. So we had one experiment, and within our experiment there was numerous subjects, and then each subject will have a few sessions. Within each session, there'll be several runs, and then each run has a series of brain volumes, and then each volume has multiple slices, and then each slice will have many voxels. And each voxel will have one intensity associated with it, which means there's a lot of data acquired during this process. So on your left, there's an image of a matrix, and that blue box represents one voxel. And just to give you an idea of the size of it, for pre-processing data, it could be upwards of 1 gigabyte, and then for post-processing data, it could equal 3 gigabytes or more. But that's just the average per session. So if you're having multiple sessions within a subject, then your data size will increase. So this is the bladder filling protocol. So subjects are catheterized and connected to a urodynamic pump that mimics the bladder filling and emptying. So there's infusion of saline and withdrawal of saline. So prior to that, we take a structural MRI scan and then 2 resting state scans. Where their bladder is empty, and then we fill their bladder with 100 milliliters of saline, and then these are 2 empty blocks, and each block will have 4 runs. And within the runs, there's a saline infusion and withdrawal phase that is happening right here. So in these 2 blocks, there's one task-based fMRI scan taken with an empty bladder, and then the second task-based MRI scan with an empty bladder. And then after that, we'll fill their bladder until the subjects have a desire to void, and then they'll let us know. And then the full bladder resting state is taken, and then followed with 2 more blocks of task-based scans, but with a full bladder. So again, those blocks have 4 runs each. So this is the Adolescent Brain Cognitive Development Human Connectome Project Pipeline. We just use acronyms. So it's ABCD, HCP, fMRI pipeline, makes it easier to say. So the DCAN lab, which is the Developmental Cognition and Neuroimaging lab here at OHSU, has a modified version of that they use. And this is a minimal pre-processing pipeline that we put our data through just to remove less information and keep as much data as possible. So it brings multimodal data together to be compared across subjects. For this particular study, we're combining modalities of T1 weighted image, T2 weighted image, the resting state, and the task-based scans. So the input for that is brain imaging data structure, which is something that the DCAN lab uses. So their pipeline has a BIDS initiative built on top of the HCP pipeline just to make configuration easier. And then the output is a Connectivity Informatics Technology Initiative, which is called a CIFTI. And within the HCP pipeline, you can use special pipelines options. So if you're working with a specific subject or like specific population, such as an older population or a neurodegenerative, you can input a template and that will make your results better. And then the ABCD quality control is a standard operating protocol that the DCAN lab uses to assess the quality of all the images acquired. So you're going to assess both the structural and functional scans, and these are rated on high quality, medium quality, or low quality. So this is an example of structural quality assessment. So on the left side, I have quality assessment of 1, which means that they're high quality scans. And then on the right side, I have quality assessment of 3, which means they're the lower quality scans. So what you're looking for is gray matter, which is the red lines. It's a little blurry, but I promise you the red lines are there. And the white matter, which is the black lines. So you want to make sure that they're not intertwining with each other, and then you can clearly tell between the gray and the white matter. So you can see that they're delineated on the left side, but on the right side that there's some distortion up here at the superior parietal, and then on the right temporal there's also some distortion, which means that it's going to receive a quality assessment of 3 because you're not going to want to use a scan with some distortions in it. And then this is a functionally quality assessment. So again, I have the higher quality images on the left side and then the lower quality on the right side. So the top row is the structural overlaid on the functional, and then the bottom row is the functional overlaid on the structural. So for these ones, you're looking for any dropout or field of view errors, and again, for distortions or warps of the brain. On the high quality images, there's some minimal temporal dropout right here, but that's fine because it's very minimal and it's not affecting the brain at all, the brain imaging. But on the right side, you can see that there's some field of view error in cutoff of the cerebellum, the occipital lobe, and temporal. So we would not use that scan for analysis because a lot of the brain was cut off during imaging. So this is a post-regression Granites and motion numbers graph. So we use the post-regression because it shows the whole regression of the brain, and we're looking to pinpoint motion points during data acquisition. So on this side, this is the frame displacement graph right here, and then this is the gray ordinates graph. And you're going to want to look at both of them when you're determining if the subject has moved too much during data acquisition. So when you're looking at frame displacement graph, there's a threshold that is usually determined during analysis, and in most cases it's a 0.2 threshold. So right here, the yellow line is the 0.2 threshold, and you want a majority of your frames to be below that line. So on the left side, you can see a majority of the frames collected is below that threshold, whereas on the right side, a majority of it is above that threshold. And the vertical static bars indicates motion during data acquisition. So on the right side, you see that the subject was moving a lot, and there's a ton of those vertical bars. And that tends to happen in the cases with urgency incontinence, because when you fill their bladder, they're uncomfortable and they're going to move a lot, and they can't stay still during the MRI. Whereas in the subjects with no history of overactive bladder, they're fine and they aren't uncomfortable with the full bladder. So this is just an overview of quality assessment. So this is the total number of scans that we acquired in this project. And then, as you can see, a majority of the scans that we collected were of high quality, so I divided it up as functional rest, functional task, and structural. And a majority of them are in the one category, which is high quality, and there's very few that are of low quality. So for next steps is to decide the quality cutoff for inclusion. So we would determine quality thresholds and then test out those different quality thresholds to see how many subjects are taking out, depending on how strict that threshold is. And you also need to keep in mind how many subjects you want to move forward for analysis and what type of analysis you want to do. So we were going to do an FCON ANOVA, which is a group comparison of brain activity. So we would compare the brain activity of the subjects with urgency incontinence versus the subjects with no history of overactive bladder. And then the next analysis would be the FCON regression, which is association with symptom severity. So we would be looking at the validated questionnaire scores that were collected in the beginning, as well as their bladder diaries, like how many frequency, urgency, or urge leaks, nocturia episodes, and fluid intake they had per day. Some internship takeaways. So I learned the importance of computational data organization very early on, especially when you're working with big data sets. So when you're using multiple pipelines or tools or graphic user interfaces, you should have input and output folders for each of those tools, because you're going to be inputting a different form of that data or file type into that each pipeline. And then I also learned that you should be naming everything very descriptively, just to go back on, and it'd be easy to find what you're looking for. And then to always do test runs and proofread. Everybody that I worked with at the DCAN lab could not stress this enough in using the echo command, because when you're executing and bash scripting your data, you don't want to do anything that could make some irreversible damage to your data, and it's always just good to test it out first and proofread and make sure you don't have any little typos that will make the command run. And then I learned that patience is key when studying brains and working with newly developed tools. So this summer, I did a lot of navigating and programming in the command line as well as cloud computing. And I was working a lot in Exacloud, and every time I would get someone to help me, they'd be like, oh, what are you working in, like Rushmore or the ARC? And then I'll be like, oh, I'm working in Exacloud, and everyone just sighs really loud. Because it could be really slow, and there's a lot of problems with it. Like, for example, I could run the pipeline on one day, and then the next day I'd do the exact same thing, and it won't run. And I don't know why. And then the next day, it'll run perfectly. So it's just understanding that studying brains and working with advanced technology is going to be difficult, and it's going to take time. And then to take advantage of experts in the room. I was lucky enough to have a desk in the DCAN lab, which is essentially a huge room with just a bunch of computers. So I sat around their computing team, and whenever I needed assistance, help, or like any questions, I knew that I could just get up and like ask my neighbor, like, hey, I need help, and they would come over and help me out. So I'd just like to acknowledge Dr. Karstens for taking me on this summer, as well as her collaborators, Dr. Damien Fair in the DCAN lab, and then Dr. Rahel Nardos, and then DMICE, and then Developmental Cognition and Neuroimaging lab that I worked in, and the National Library of Medicine grant that made this internship possible. Thank you.
I don't know if this is working.
It's on.
Okay. My name is Peter Graven. I am a data scientist in ITG with OGSU. We had for the summer, Dave Dorr is also a member of the internship project that Ian Schipper has been working on. So we're excited to have him. He's kind of the first intern in this data science and advanced modeling that we've been doing. And so it's been great experience for us and hopefully for him as well. We kind of played around with a couple of different possible topics for the summer that were related to projects we had that were necessary, and he really gravitated onto wanting to create his own predictive model and test some of the new techniques or whatever that he was interested in, and getting comfortable with that. So it's been a great experience. We've liked having him, and hope to do it again in the future. Go for it.
Major at St. Olaf College, and we're going to be talking about the use of machine learning and artificial intelligence in healthcare, specifically in risk stratification. So just a quick overview of what we're going to be doing. We're going to talk about why this project in particular and why we're looking at such a specific subset of patients. We're going to talk about the data that we've been working with and a lot of the features of it, as well as the cleaning and formatting, which was a whole task and a half. Then we're going to get into some modeling, the results, and then where this project's going to go in the future. So just a little disclaimer, this project is very much in the works. All the models I'm going to be talking about today are actually old models. We've got new ones that are running right now on my computer down on the waterfront. And so it's still very exciting. And so without further ado, just a brief overview of who I am. That is from Les Mis, the song, Who Am I? Yeah. Anyway, so just a little explanation of why a music major is sitting here talking about AI. I'm also an economics major. I do most of my work with healthcare economics and predictive modeling, done a lot of work with billing, and so it's nice to get a bit of a change of pace and work a little more patient side. So that's a bit why I'm here talking about AI and not something a little more appropriate to music, such as the eye surgeon who managed to kill both Johann Sebastian Bach and George Friedrich Handel. And I promise that is my only music fact for this presentation, so that should save you some distress. So this has been a great project with a great team. Myself, I'm the intern. I've been working mostly with the modeling as well as some data formatting. David Dorr and Peter Graven were both my mentors in this. David Dorr did a lot of informatics work as well as talking us through some of the work that's been similar to this, since he's done quite a bit of it. And Peter's been kind of coordinating the whole effort and getting us through what kind of models we need, what models we want, and where we're going to go with this. Conrad has been doing a lot of work with statistics. He's been handling a lot of the data cleaning and formatting because it is a lot of work that I am, truth be told, not super great at. And so he's been helping a lot with that, as well as with logistic modeling. And then Faiza Khan and Ben Orwell both worked on retrieving data from Epic and helping us kind of understand different clinical aspects, since Peter, Conrad, and I are all either statisticians, data scientists, or economists. So we don't have a whole lot of clinical knowledge. So if you ask me what is a normal value for a creatinine lab, I would just smile and blink at you until you told me. So that's kind of who we're working with here. So this project, risk stratification, has a lot of uses, especially with predictive modeling, and we can take a patient population that is at particularly high risk of having an adverse outcome. This could be something like going to the emergency room. This could be incurring exceptionally high costs, which is in excess of $30,000, or this could be being hospitalized. So these are all things that we would like to avoid, and we've actually we have been introduced to a specific intervention that we would like to use, which involves, it's called tablet-based patient monitoring, where we send particularly high-risk patients home with a tablet, and so they can continue the monitoring process that goes on in the hospital, at home, during their outpatient treatment. And this is particularly important for cancer patients, because cancer patients have a whole host of other problems that most patients don't have to deal with, especially if they've undergone chemotherapy. Because chemotherapy affects the body in a whole bunch of different ways. It can compromise your immune system. It can cause severe nausea. And so there's a lot of problems that could seem very severe, but might be perfectly normal for a patient with that cancer and that treatment, or they could actually be incredibly severe. And so this tablet monitoring gives us the ability to have them report in to their doctor and say, this is what's going on. Is this normal? And so, yeah, that's what we've got. We have about 20 tablets in total that we can send home. And as we'll go over later, we're talking about a patient population that we're talking about 83,000 people. And so we can only send 20 home, and so we want to be really sure that the 20 we send home are with the right people. And so it does have kind of limitations of resources, and so we want to make sure the right people get these. Okay, so the data. Data comes from Care Everywhere, which includes OHSU and other organ healthcare providers. And so we're primarily concerned with only the patients who have had an office visit and are undergoing treatment at OHSU. And so that's one of the ways we pared down. I think we started with somewhere around 90,000, and we cut about 7,000 patients who did not receive their treatment at OHSU, but those 83,000 may have gone elsewhere for an emergency room visit or a hospitalization, and so we want to be able to capture that even though they are an OHSU patient. And so we have, I think, 7 different data sets that we pulled from. These are data sets of office visits, ED visits, hospitalizations. We have 2 million lab results of 2,000 independent labs. We have their demographic information. We have more patient information. It is a whole bunch of stuff that we had to pair together. And so the way we did that is every patient has a patient ID, and every encounter has an encounter ID. So let's say a patient goes to the emergency room. That visit will have its ID, and it will have their patient ID. And then let's say they get a lab done. That lab result will go into the lab data set under their patient ID and encounter ID. So we can say this lab came from this. Let's say they get diagnosed with a certain disease. That diagnosis code goes into its own data set with their patient ID and encounter ID. So you can see we're kind of pulling from a lot of different places to create a single data set. And so the final data set had 83,913 patients with 212 different variables. These variables are primarily 0-1 indicators of do they have this diagnosis or not? Do they have this lab result as a high or not? Do they have this cancer or not? And so that makes it a lot easier when we come to modeling. So yeah, we've got 83,000 patients in the year 2018. We'll be predicting their ED visits in 2019 eventually, but the models that I have are all predicting on their 2018 data sets in order to see, can we accurately predict their ED visits based on all of this data? And we have a bit of a problem in that very few of the cancer patients that we have actually went to the emergency department in 2018. It's about 6%. And so if you have a naive model, which is a model that only predicts one thing, you could just say, no one's going to go to the emergency department, and you'd be right 94% of the time, which is a really, really good model. And so we need to be able to beat that. And there's a couple different ways we can do that. And then there's a few ways that we can get lower accuracy, but be able to catch a higher number of the patients we're actually interested in, those high-risk patients. So now we're going to get into a brief overview of the modeling that we're doing, the techniques that we use. I'm going to go really quickly over logistic regression because I'm not going to talk about any of the logit models since I didn't make them. That was all Conrad's work. Then I'm going to go over random forest classification and artificial neural networks. So logistic regression is probably the one that most of you are at least somewhat familiar with. This creates a linear function to predict a probability or a log odds of a 0 or a 1. And so you can come out with 0-1 indicator of, say, are you going to go to the emergency department? And the advantage of these is that they're pretty computationally inexpensive. The average logit model takes, for me, about 5 to 10 seconds to run. And that's going to be a marked difference that we're going to see later. But they are actually rather simple, and they're good for simple relationships. But if you have a nonlinear relationship, or if you have something that just, it's more complicated, they can tend to struggle. And they're also very prone to overfitting, where if you include collinearity, it's not quite robust enough to deal with that. And so we get to the random forest models, which are kind of the next step up in being complicated, and it's quite a big step. So what these do is these will create a number of decision trees. And this little graphic over to the right is actually a pretty good idea of what each decision tree looks like, where you start with a number of instances of inputs. It'll undergo a bunch of logical functions in order to come out with a single output. And so the average random forest has about 500 trees, and it will run all 500 of those trees on every observation. So if we're looking at a data set of 83,000 people, times 500 trees, we've got about 45 million trees, which is more trees than there are in the state of Oregon. So these are big models. They are hard to understand, and they get out of hand very fast. And so the average model that I had would take about 20 to 25 minutes to run. I've had them go as long as 2.5 to 3 hours, or if you have a particularly slow computer, a weekend. And so these are really useful for more complicated relationships. They handle nonlinear relationships incredibly well because it handles each instance of a numerical value as its own thing. So it separates, say, a 0 to 10 into 10 different points that could each have a nonlinear relationship to your output. And the final one is the artificial neural networks. Fortunately for, I think, the human race, I'm not super great at making a thinking machine, but I can make one that can do a binary classification pretty dang well. And so what this does is essentially attempts to replicate the way the human brain works. You have a layer of input neurons, which will have values. So for each variable, you have a different neuron. Those will undergo then a layer, several hidden layers of neurons, which include logical functions, in order to say, okay, is this true? Then you're going to go over there. Is it false? You're going to go over there. And so it undergoes all of that until you get to a single output layer. So I only did one artificial neural network for this because they are quite complicated and quite computationally expensive. My model had about 3 hidden layers. The general rule of thumb is each layer will contain 2/3 the number of inputs of neurons as the previous layer until you get down to a single one. And yeah, these are very, very, very expensive. The one that I made took about 48 hours to run constantly. I kind of left it there for a weekend and came back, and it was still chugging away. So these are not the greatest if you don't have a whole bunch of time. And we're going to run into an issue later of importing these into the reporting workbench for use in Epic. So now that we know kind of what the models are, we have a few struggles. Machine learning models do exactly what it sounds like. They learn from the data. And so if you don't have a lot of positive instances of something, they can have trouble learning from that. So in this case, we only have 6% ED visits. And so it learns ED visits are very, very rare, and they're probably not going to happen. So I'm best off just hedging my bets and saying you're not going to go to the emergency department. That's not what we want. And so there's kind of 2 ways that we can do that. We can change function parameters, or the way that I chose to do, we can change what data it's trained on. So what I did for that is I created a separate data set that has 2,000 observations, 1,000 of which did not go to the emergency room, and 1,000 of which did. So I tricked the model into thinking emergency room visits happen about half of the time, and so you should learn what causes people to go to the emergency room. And this does cause it to suffer in terms of accuracy, but as we see, it'll help us in sensitivity. And so accuracy is, while a great metric, is not the one we're primarily concerned with. We're concerned with positive predictive value and sensitivity. So positive predictive value tells us, of the things that we have classified as positive, how many were actually positive. So this is our confidence that if we say you're high risk, you're actually high risk, which is important because we only want to give tablets to those who are high risk. And then sensitivity, which is how many of the high-risk patients did we actually catch. So I'll be talking about 5 models. We have a full representative model, which uses all variables at our disposal and uses a representative set of the data. We split our data into a training and testing set to avoid overfitting as much as we can. 80% of our data was in the training set and 20% in the testing set. And so that was trained on the full 80% training set. A full even model, which uses all of the variables and uses my special even data set in order to condition it. A reduced representative model, which uses a reduced subset of variables. And the way we reduced those is we looked specifically which HCC diagnosis codes are most highly correlated with instances of emergency room department. And we took the top 10 of those and included those in our model. And we did the same for LDAs, which are lines, drains, and airways, and for labs and for cancers. And we used a set of demographic information. So that was trained on all of the data. The reduced used the same set of reduced variables and used the altered even subset. And then my artificial neural network, which I call AN, used a further reduced set of variables because artificial neural networks can only handle numerical or complex vector arguments. And so if you try to put in a categorical variable, it will just cry and give up. So the results, I listed out the true positive, true negative, false positive, and false negative. If you're interested, we can see our accuracies are pretty good. The 2 full and the 2 representative models have above that 94% accuracy, which is exactly what we're looking for. So these are doing better than a naive model. But we can look at their sensitivity and see it's about 10%, which is pretty bad. And their positive predictive values are not what we would like either. The even, on the other hand, have a slightly lower accuracy, but still pretty decent at 82%. But we can see their sensitivity gets much better with 61% accuracy and 58% in the other case. And then the artificial neural network has slightly worse accuracy, slightly better sensitivity. It's just kind of a nice middle-of-the-road model. So going forward, I mentioned integrating these into the reporting workbench. What that is in Epic, we can put in a model with all of our data manipulation in order to turn raw input from physicians into an output of a prediction of a risk. And there's a few problems here that we're going to run into, and that is what the Epic servers are programmed to handle and what they're compatible with. So they're compatible with all linear models, all logistic models, and simple random forest models. And so notably, artificial neural networks are not yet programmed to be put into the reporting workbench. So unfortunately, Anne is kind of useless to us at this point. And the more complicated random forest models are just not ready to be put into the Epic workbench because they're too complicated, they're too computationally expensive, and doing so would just bog down the whole system. And once we do that, we'll move on to patient trials in which they'll actually be used, and we'll say, okay, which are the highest risk patients, and we'll send them home with tablets, and we'll see if that actually helps them. And hopefully it will. So some thank yous. The National Library of Medicine Training Grant, of course, the Department of Medical Informatics and Clinical Epidemiology, as well as Peter Graven and his team, and the Care Management Plus team down at 3030 Moody, who have so graciously accommodated me in their office, even though I do some pretty different work from them. They've been great colleagues and helped quite a bit with both my presentation, poster, research techniques, and the like. Thank you.
Hey, thank you. Great presentation. One specific thing I was wondering about, you said that there are only 20 tablets. Have you tried looking at what happens if you give like the top 20 highest risk? Well, if you pretend that you had done that with the 20 that were labeled as being highest risk, and you see how many of them actually did end up coming back to the ED, how many of them did you miss?
Yes, thank you. Actually, I forgot to mention that. We did look at, of the top 20 that the model classifies, which ones have the highest accuracy. I didn't include it here, but the accuracies were much lower than what it stated in the accuracy for the complete model, but still on the higher side, between 50 and 75% for all of them, which it's up to the institution if that's an acceptable level of risk of catching 25% not actually high-risk patients. And so that's up to people who are not me. But it does have some promising aspects to it.
Which model's the best and why?
It really depends on your thought process of what you prioritize. If you prioritize accuracy above all else, the reduced representative model is going to be your best bet, 96.7% accuracy. If you're valuing sensitivity, so catching the most number of actual high-risk patients, for instance, if we have, say, a larger number of tablets in the future and we want to make sure we help as many of the high-risk patients, I would say that the full even model would be the best because it catches the highest percentage of high-risk patients. So it depends what you prioritize.
Well, it seems like you have a metric because you only have 20 tablets right now to do the study with. So it would seem that the right metric would be, the precision at 20, which is, how good.
In which case it would be the full representative,
okay.
75% accuracy.
And you said you get, so at 20, I guess all the predictions are positive?
Yeah, so all the predictions are positive and 15 actually went to the emergency department.
So 75% accuracy, that's probably pretty good. I don't know. Peter can give you an economic analysis if that makes it worth doing the study, but it sounds like that would be reasonable to me.
Compared to the baseline minus 60%.
Right. And would any of these other models even get close to that?
The reduced representative had a little over 50%, which is not bad. Again, better than sticking a cat and saying, here, decide who gets a tablet. But ideally, you'd want the highest accuracy.